Hello friends, welcome to another video tutorial from Shomo's Biology. We've been talking about immunology video lectures for a quite long time. In this video, we'll be talking about antibody structure and function. So we'll see the different classification of antibody and their function a little bit. So let's start. First of all, what is antibody? Antibody are peptide molecules that are secreted from plasma cells. So let me write it there. What happens actually in our body, there are two different modes of immune system. One is the cell mediated immune system, another one is known as the humoral immune system. Among humoral immune system, we have things like complement and we have antibody mediated immune system. The thing about the difference between cell mediated and humoral immune system is in cell mediated immunity, it is the first line of defense, like the first line of immune defenses that we see. Like there are macrophage, dendritic cells, there are natural killer cells that protect us against our infections. If there is any infective agent, they will kill that or engulf that. But once the interaction with such of those antigen presenting cells involved with the T helper cells, they activate B cells. And once they activate B cells, this B cell convert itself into plasma cell. Plasma cell are bigger than B cells. This small modification take place in the B cell. So it have a huge area of cytosol where they start producing different types of proteins known as antibody. Now these proteins are very specific against a foreign pathogen known as antigen. Now the typical structure of antibody is this Y shape. Hope you see this structure earlier. And this is kind of exact structure because it have two heavy chain and two light chains. Now with the help of this specific Y-shaped structure, it has a region called FAB, which is this hands of Y. These two regions are known as FAB portion. With the help of this FAB, they can specifically interact with antigen. Okay, and there is this portion that is in the bottom known as FC portion. With the help of this FC, they involve and interact with the cell surface receptors of our immune cell. For example, B cell have this B cell, this receptor. With the help of this FC region, antibody can easily bind with that receptor by this fashion. So you will see a lot of antibody attached to the surface of B cell with the help of these FC interactions. Now let's look at the structure in a little bit more details. If you look at here, this whole chain is a heavy chain. This is also a heavy chain. And the small fragments are known as the light chain. Now the chains are very same type of chains repeated time to time. And for different class of antibody also the number of chain varies. Not the number of chain but the repetition of the amino acids in the chain varies. And also those different zones of the amino acids are attached with themselves by the disulfide linkage in different regions. You will see huge number of disulfide linkage. For example, if you just divide it in a further class, you will see there is something written as V and written as C. The first fragments which are in direct contact with antibody, as I discussed earlier, like if this is the antibody, the FAB portion is always in contact with the antigen. That portion is known as a variable region or V region. Variable region means the amino acid sequence of this region can vary from cell to cell, from time to time. And that is very important because in our body there are so many different pathogens are infecting us, so many different pathogens are involved in the pathogenic reactions. So our body needs to be prepared against all of them. We don't know what kind of antigen is going to enter. So whatever type of antigen enters, we have at least one match with the help of that variable region. So it's our body's job to produce as much as variety they can produce in the production of antibody. Small amino acid changes, mutations known as a hypermutation take place that helps in changing the amino acid sequence so that a little variations can also change to interact with different array of antigens that infect us. Okay, and the rest of the region is known as C for constant. C for constant region, because those part of the amino acid sequence remain constant. They don't change, mutation don't take place often in those regions. Okay. So you see here the statistics, that 2 light chains of each 25 kilodalton, 2 heavy chains of it 50 kilodalton. Now these light chains and heavy chains, the heavy chains are identical to each other, light chains are also identical to each other. And each of these light chain are attached to the heavy chain with the help of this disulfide linkage here. So we have a linkage between heavy chain. Here are the two disulfide linkage together, and we also have linkage between heavy chain and light chain with one disulfide bond. We also have intra disulfide linkages between the amino acids, as you can see in this picture. So that overview about the antibody structure. Now let's talk about the antibody classification based on their functionality. Here I am not going to talk about the structure of each of the type of antibody. They are classified based on their function as well as the number of repeats of number of repeats of amino acids they have in them, and also they have different properties based on the number of amino acids that are present there. For example, if we begin with IgA, antibodies are also known as immunoglobulins because they belong to the this globulin family proteins. So IgA or immunoglobulin A is this is a single unit of this antibody. It usually, when it's secreted, it combines with two such subunits and form a complete unit of IgA. A complete unit of IgA contains two antibodies together linked via a joining chain or J chain. IgA is also called as secretory antibody because it's very often found in our saliva, in our tear, in the breast milk, and in different mucous membrane or mucosal layers. Okay, so it protects us against pathogen in all this case. That's why we tell that in the eye, the water of a tear as well as the breast milk, all these things contains a little amount of this antibodies, and it has a immune response against the pathogen and the response to fight against the pathogen. Second type is the IgD, immunoglobulin D. Immunoglobulin D, the structure is very similar. The single unit of immunoglobulin D, the structure is similar with the IgA as well, but this immunoglobulin D is present as a single molecule. They don't have any dimer or something. Immunoglobulin D is responsible for interacting with mast cells and causing allergic reactions in our body. Okay, this is not a kind of good job, but still its activity is to involve with some mast cells. Let's say this is a mast cells with the surface receptor, and they have all those mast cells are filled with all those vesicles. Vesicles are filling with all those enzymes. Now once IgD is present there, it can involve with that. It can also cause some sort of hypersensitivity reactions as well, and autoimmune reactions as well. Third one is IgE. IgE also involved in the process of allergy formation. It's also involved with the process of hypersensitivity, especially type 1 hypersensitivity. It also can interact with the receptor of the mast cell with the help of this Fc region, and once two such IgE involved, it can fix that allergen that can provide some signal inside the mast cell, which will allow the cell to release some chemical granules and factors outside that can cause some serious damage to our tissue. Like those chemical factors could be histamine, leukotriene, and also bradykinin, and so many things that ultimately can cause anaphylactic reactions like airway constriction or vasodilatation or things like that. That's the part of immunoglobulin E. But a good thing about immunoglobulin E is that immunoglobulin E or antibody E can also fight against worm. Let's say worms are huge compared to these proteins or any other immune cells. They are large pathogens. So what immunoglobulin E can do, let's say this is a worm, immunoglobulin E can interact and coat the worm all over. Once it coats the worm all over the places, then it will bring some cytotoxic cells into the place, because once the worm is coated by immunoglobulin E, this worm is suddenly become more delicious for our cytotoxic immune cells. So cytotoxic immune cell can come now and can attach to this FC region of this immunoglobulin E, and they can start release some chemical factors that can create pores in the membrane of the worm that can ultimately kill the worm. So it's a very important job it can play in this case. It is known as antibody dependent cellular cytotoxicity, and some cases they also bring neutrophils or eosinophils in this place. Eosinophil is a type of white blood cells in our body. Eosinophils can interact with this FC region and start releasing chemical factors that can kill the worm. So they involve in this process, though it's not exactly as a statement, because this whole process is kind of prescribed and under investigation. We have immunoglobulin G. Immunoglobulin G is a type of antibody that can be transferred from one to the next, because this is the first type of antibody that we have as a child, as a newborn baby, because immunoglobulin G has the capability of transferring through the placenta into the newborn baby. That's a unique feature about immunoglobulin G. And the job of immunoglobulin G as well is to present like on the surface of the bacteria B cell, as well as once these B cells are ready to make antibody against specific antigens, they make more and more immunoglobulin G. So immunoglobulin G are very active against antigens. We use them for a process known as opsonization. Opsonization means where the thing where the pathogen is coated with antibody, suddenly that pathogen gets more attractive for all those phagocytic cells. That's known as opsonization. That's done by immunoglobulin G quite often. That's the job of immunoglobulin G. Immunoglobulin M, that is the last one that we want to talk, IgM. Immunoglobulin M is present as a pentamer. Okay, so 5 such immunoglobulins joined together with joining chains and disulfide linkages that forms a pentamer. And the idea of immunoglobulin M is simple. Immunoglobulin M is the default immunoglobulin provided or produced by any B cell, and it's also the most common form of immunoglobulin that are found on surface of B cells. So if you look at B cell, you see B cell receptor. They are nothing but these immunoglobulins, or IgM, act as a B cell receptor on the surface of B cell. Now this immunoglobulin M can form this pentamer, so it gives them a unique feature of fixing complement in higher order, because in fixation of the complement we require for the complement protein to be in contact with immunoglobulin. So the more the immunoglobulin is spread, the better the contact between the immunoglobulin and complement. That is what we want to do, right? So immunoglobulin have 5 different chains. That's why we can use that for fixing complement. So immunoglobulin M is used for fixing complement, and it helps in fixing complement in a much faster way compared to the rest of the immunoglobulins. Otherwise, the job of immunoglobulin is it's a default form. So normally if there is no signal from the T cell to the B cell, then the B cell normally start producing immunoglobulin M. Now if there is a unique signal, then they shift from producing immunoglobulin M to immunoglobulin G or any other type of immunoglobulin. That's the idea of this IgM. Now if you look at the function, one function of antibody are neutralization. Neutralization means if there is any pathogen or fragment of the pathogen which is known as antigen, which is depicted here with this red color, if this thing enters, antibody can bind with that antigen and form an antigen antibody complex. It is called as a neutralization because the antigen is not free, it's masked by the antibody. It can also cause opsonization. Opsonization is while a full pathogen take entry and that pathogen is coated by antibody so that phagocytic cells can easily engulf that pathogen inside and can kill it. That's called the opsonization. Normally, if there is no antibody, pathogen is present, phagocytic cells still can engulf it and can destroy it. But once we coat the pathogen with antibody, suddenly that becomes a delicious meal for the phagocytes. So phagocyte can engulf them much eagerly after this antibody coating. Sometimes this antibody can also involve in the signaling process. Here Ig subclass 4, because I haven't talked about that. These immunoglobulins also have subclasses in them. So this IgG, one type of IgG antibody, they can involve in some sort of chemical signaling inside. That can also possible. And also this immunoglobulins are also involved in the process of simple interactions, and they also act as a receptor in surface of B cell. Okay, so they are also involved in cell signaling. Immunoglobulin can involve in the complement activation pathway, where you see once the immunoglobulins are attached with the surface protein of the target pathogen, complement molecules can interact with the Fc region of the immunoglobulin that can fix the complement, means complement can be activated, and so many complement proteins finally can create pore in the membrane of the target cell that can kill that pathogen. And antibody is also involved with the process of antibody dependent cellular cytotoxicity, or ADCC, where the T cell can recognize the Fc region of the antibody and it can start releasing chemical factors that can kill the cell by cell lysis. Obviously the T cell we are talking about is a cytotoxic T cell here. Okay, that's the same type of thing that we talked about in the immunoglobulin E and eosinophil mediated worm killing, right? So that's all about the structure and function and classification of antibody. I hope you understand the video. If you like this video, please hit the like button, subscribe to my channel to get more videos like that, and definitely share this video with your friends. Thank you.